Phase contrast microscopy, first described in 1934 by Dutch physicist Frits Zernike, who was awarded the Nobel Prize in 1953, is a contrast enhancing optical technique that can be utilized to produce high contrast images of transparent specimens such as living cells, microorganisms, thin tissue slices, and subcellular particles. Two or more rays of light emitted from the same source with the same frequency are referred as coherent rays. Two such rays of equal amplitude in the same phase are able to combine, and hence the amplitude doubles. Therefore, the light is twice bright. If, however, they are out of phase or in reverse phase, they cancel each other and the result is no light and hence total darkness. The phase of light rays changes when they pass through a specimen to be observed under the microscope, and this change is due to the difference in the depth and density of the cells and its internal parts. Since the refractive index of the cells and their organelles is slightly higher than that of water, a light ray that enters them will be retarded in comparison to one that does not. Additionally, the amplitude of these rays is reduced slightly since the organelles absorb and scatter some of the light. It is this difference in amplitude that allows the visualization of cells in bright field microscopy. Since light passing through cell is retarded, the cell appears darker than the background. However, the amount of light absorbed or scattered by individual bacterial cells is quite small, and hence the cell is virtually transparent. Unstained specimens that do not absorb light alter the phase of the diffracted light by approximately 1/4 wavelength as compared to the undeviated direct light passing through or around the specimen unaffected. Unfortunately, our eyes as well as camera film are unable to detect these phase differences. The result is that the image at the eyepiece level is so lacking in contrast as to make the details almost invisible, as you can see in this image on your screen. Let us now discuss the principle of phase contrast microscopy. Zernike's method was to speed up the direct light by 1/4 wavelength so that the difference in the wavelength between the direct and deviated light for a specimen would now be half wavelength. As a result, the direct and diffracted light arriving at the image level of the eyepiece would be able to produce a contrast. This is called dark or positive phase contrast. Positive phase contrast reveals medium to dark gray images against lighter gray background. These images often have a bright halo along the edge of the image. Another possible course, much less often used, is to arrange to slow down the direct light by 1/4 wavelength so that the diffracted light and direct light arrive at the eyepiece in phase. This is called negative or bright contrast. Negative phase contrast is opposite. Specimen appears lighter against a dark background. They also have a halo outlining the image. To develop a phase difference, it is necessary to separate the light beams interacting with the specimen from those that do not encounter the specimen. This is obtained by two additional plates, namely annular diaphragm and phase shifting plate. Let us talk about the annular diaphragm first. The annular diaphragm is typically constructed as an opaque, flat black plate with a transparent annular ring which is positioned in the front focal plane of the condenser so that the specimen can be illuminated by defocused parallel light wave fronts emerging from the ring. As the hollow cone of light from the annulus passes through the specimen undeviated, it arrives at the rear focal plane of the objective in the shape of a ring of light. The fainter light diffracted by the specimen is spread over the rear focal plane of the objective. To speed up the direct undeviated zeroth order light, a phase plate is installed with a ring-shaped phase shifting plate attached to it at the real focal plane of the objective. The phase plate contains a ring etched into the glass that has reduced thickness in order to differentially advance the phase of the undeviated wave by a quarter wavelength. The phase shifting plate is mounted in or near the objective rear focal plane in order to selectively alter the phase and amplitude of the undeviated light passing through the specimen. Unstained specimens that do not absorb light alter the phase of the diffracted light by approximately 1/4 wavelength as compared to the undeviated direct light passing through or around the specimen unaffected. When this deviated ray passes through the phase shifting plate, it is further retarded by 1/4 wavelength, making it now half a wavelength out of phase as compared to the undeviated ray. The deviated and undeviated rays cancel out each other and increase the contrast. Let us now understand the working of the phase contrast microscope with the help of this ray diagram. In a phase contrast microscope, when a single ray of light falls on a specimen, two rays develop. One of these is the direct or undiffracted light ray from the annular diaphragm. This ray passes through the specimen and is then focused on the phase shifting plate. The phase shifting plate will either delay or advance this ray. The second ray passes through the margin of the specimen and hence is scattered or diffracted. This ray does not strike the phase plate and hence its phase is not affected. When these two rays, the direct, which is red in color in this picture, and diffracted, which is blue in color in this picture, unite. They are not in phase and the phase differences become apparent. Two sets of rays emerging from the same point of the specimen return to the same point in the image but have different phases of half a wavelength with respect to each other. This results in variation in intensity of illumination and therefore contrast is produced. As a result, the specimen appears darker against a light background or lighter against a darker background. I am now going to show you two images. One bright field image and another the phase contrast image. You can see that the phase contrast image shows minute details like cilia of the unstained specimen, which is impossible with the bright field microscopy. Let us now move on to the applications of phase contrast microscopy. Phase contrast is preferable to bright field microscopy when higher magnifications like 400x and 1000x are needed and the specimen is unstained or the details so fine that color does not show up well. Phase contrast microscopy enables visualization of cilia and flagella which are nearly invisible in bright field microscopy but show up in sharp contrast in phase contrast microscopy. Phase contrast microscopy enables visualization of protozoans. Amoeba look like vague outlines in bright field but show a great deal of detail in phase. Most living microscopic organisms are much more obvious in phase contrast microscopy. Phase contrast microscopy enables the researcher to see otherwise invisible structures like nucleus found within the cells. Phase contrast microscopy is extensively used to observe living cells in tissue culture to monitor their growth. These cells are not very clear in bright field as you can see here. Phase contrast microscopy enables visualization of chromosomal movement during cell division. Phase contrast microscopy is used to observe phenomena like phagocytosis. Phagocytosed E. coli are visualized inside vacuoles known as phagolysosomes shown as white arrows in this picture. The phase contrast microscope has many advantages as compared to that of bright field or dark field microscopes. First, living cells can be observed in their natural unstained state. Highly transparent biological cells are more clearly visible. No special preparation of fixation or staining is needed to study an object under a phase contrast microscope, which saves a lot of time. Also, it avoids damages of cells due to chemical preparation and staining. Phase contrast microscopy allows examining intracellular components of living cells at relatively higher resolution. It made it possible for biologists to study living cells and how they proliferate through cell division. Phase contrast optical components can be added to virtually any bright field microscope provided the specialized phase objectives conform to the tube length parameters and the condenser will accept an annular phase ring of the correct size. The cost of phase contrast microscope is affordable. Live cell imaging and cell processing is possible. Optical construction of a phase contrast microscope is relatively simple. Let us talk now about the disadvantages or the limitations of phase contrast microscopy. The limitations of phase contrast microscopy actually are not many. Phase contrast condensers and objective lenses add considerable cost to a microscope, and so phase contrast is often not used in teaching labs but is the preferred microscope in research labs. To use phase contrast, the light path must be aligned. Generally, strong illumination is needed for phase contrast than corresponding bright field viewing, since the technique is based on the diminishment of the brightness of most objects. Phase contrast produces a halo around the images.